That's a mouthful. So this is an older paper, 2003. It's over a decade old, but I think it's really excellent for introducing these concepts, or reinforcing, depending on whether or not you've had these in class before. So here's a bunch of information that will be useful. This is a Cell paper, so it's pretty long. It's got a number of figures, but it's really excellent too. We're going to talk about epigenetics, we're talking about yeast, and something about genetics, heterochromatin in yeast. So hopefully everybody's at least heard about epigenetics. If you don't know what it is, that's okay. I stole from Wikipedia several things in this, so you'll recognize some stuff. But epigenetics, basically the study of things that are inherited from generation to generation that are not DNA sequences. Right? DNA is the heritable molecule. It's how we pass traits on from generation to generation. But we've learned in the last 20 or 30 years that it's not just A, T, G, and C that is passed from generation to generation. There are other molecules associated with DNA that get passed on, and those are called epigenetic marks. So epigenesis is the idea of, especially related to twin studies. These are genetically identical twins, why do they look different? Why do they act different? Why do they sometimes have different health problems, despite living in the same environment, having grown up in the same environment, living essentially the same lives, but one of them dies of breast cancer, the other one doesn't. There are more differences than just genetic differences. There are epigenetics. So what does this have to do with molecular biology? DNA in our cells is packed into chromatin. DNA gets wound around proteins. The proteins are called histones. This is a totally inaccurate drawing. This is not histone, this is histones. This whole thing is a nucleosome. Each nucleosome is made up of 8 histones. So there are 8 individual histone proteins, histone H2A, histone H2B, histone H3, and histone H4, that come together and make these blobs. That's called a nucleosome. It wraps DNA about twice around each nucleosome, about DNA will loop twice around the nucleosome and then go to the next nucleosome, wrap twice around it. All of this is for packaging our DNA so it doesn't break during chromosome segregation and for other reasons as well. So you get double-stranded DNA. That's basically what this is showing. Double-stranded DNA gets wrapped around histones, and then the histones get wrapped around each other and make bigger and thicker molecules, and they supercoil and supercoil, and then you get something that actually looks like what we think of as a metaphase chromosome. Highly condensed DNA. It's really tightly compacted. And that's why chromatin's really important for genetics. This is another look at that, which I think represents that idea better. The individual molecules of DNA, or the chromosome wraps around nucleosomes. Nucleosomes compact into 30 nanometer fibers. Those get coiled onto a protein scaffold. The coils coil and the coils coil. And it's amazing that the DNA can be compacted so much into something that, again, looks kind of like a chromosome. So chromosomes are quite a bit protein. It's not just DNA. Oh yes, and that's what that actually looks like. Not the smiling kids, but their chromosomes. So what we see as a metaphase chromosome is really that, it's orders of magnitude of packing of DNA. So what does protein have to do with epigenetics? Here we go. About a decade and a half or so ago, late 90s, this was all the rage in biology. Everybody wanted to study what this is. Here are the 4 histones, histone H2A, H2B, H3, and H4. 2 of each of those make up the 8 histones, so 2 H2As, 2 H2Bs, 2 H3s, 2 H4s makes up a nucleosome. And scientists started discovering that these amino acid residues, here is the first 20 or so amino acids in histone H2A. Serine, glycine, arginine, glycine, lysine, etc., from left to right. Those can be covalently modified by chemicals by enzymes. So enzymes can come along to the N-terminal tail or the C-terminal tail of these proteins and add these different chemical groups. Phosphate, you've got phosphorylation. Methyl group, you've got methylation. Acetyl group, you've got acetylation. And then UB, ubiquitination or ubiquitination. These are just a sample. This came from Wikipedia too. This is just a sampling of the different chemical modifications that can happen to histone proteins. All of these different places, there are enzymes that put all of these different chemical groups on those amino acids. This has something to do with epigenetics. This also came from Wikipedia. Okay. We now know that this is the histone code hypothesis. If you take anything away from this, this is what to remember. The histone code hypothesis. That depending on which histone you look at, so in this case, all of these are histone H3 because each of those starts with H3, right? K is lysine. 4 is the fourth amino acid in histone H3. So H3K4 means if you're looking at histone H3, you look at the fourth amino acid, it's a lysine, K. If that has a single methyl group, if that lysine is methylated, that activates expression of whatever gene is wrapped around that nucleosome. If it's got 3 methyl groups or 2 methyl groups, that also activates the gene that's associated with that nucleosome. So methylation of lysine 3, or lysine 4 and histone 3 means that you're activating transcription. You're saying make RNA transcripts of this gene, we need protein. However, and also acetylation also tends to be an activating mark, but only on these 2 residues, lysine 9 and lysine 14 of histone H3. You'll also notice that there's some methylation marks on different positions on different histones that are repressive. So the histone code basically says that different chemical groups that are on the histones can turn gene expression on or off. This is essential, and then there's that list down there at the bottom. Histone 3, lysine 9, methyl 3 is found in constitutively repressed genes. Genes that the genome says, we never want to see you make a transcript again. You'll find the histones that are associated with those genes to be methylated, trimethylated, Me3, trimethylation. So this is how epigenetic inheritance happens. You inherit not only your DNA from your parents, but also the transcription, the methylation, acetylation marks associated with their histones at fertilization. So from generation to generation, it's not just DNA sequence that gets passed down, it's also whether or not those genes should be transcribed a lot or transcribed a little bit. Are the genes turned on or turned off? It's partly, not entirely, but partly regulated by how your histone proteins have been chemically modified. And do remember, of course, that when the chromatin is packed up into something that looks like a metaphase chromosome, it's highly condensed. And in general, that means that anything that's heterochromatin doesn't generally do transcription very well because it's so condensed that proteins that do transcription, like RNA polymerase, can't actually access the DNA. The DNA is so tightly packaged that there's no way to do transcription. So another key concept, heterochromatin, really highly compressed, condensed DNA, like you see in metaphase chromosomes, and there are some other regions of chromosomes that are often found as heterochromatin. Does anybody know what the opposite of heterochromatin is? Yeah, So there's also euchromatin, which is the opposite, the uncondensed, accessible to RNA polymerase and transcription factors, and easier to do transcription. Okay, so now so that's the background, a little bit of background on epigenetics. So this paper in specific is working in yeast, and with this particular protein, SIR2. SIR2 is thought to be, before this paper, they want to know, is it a histone deacetylase? Is this a protein that controls epigenetics, controls these chemical modifications of histones by removing acetyl groups, deacetylase? If so, then this protein, by impacting how these histone proteins are modified, can thereby affect gene transcription. This protein might be responsible for turning on or turning off the expression of whole suites of genes. So they want to know, does this protein do this chemical reaction that's shown there? Is it a deacetylase? Does it take acetyl groups off of histones? So they're going to be using mutant strains. They'll be looking at wild-type yeast. They'll be looking at yeast that don't have SIR2, this protein, to see, is there a difference in anything we can observe? Does it matter? Do yeast even care if they have the SIR2 protein? And there are a couple of different techniques they're going to use to try to ask, can we see any difference between strains that have SIR2 and strains that don't? So the rest of this is mostly, here's some yeast background. Here's what yeast geneticists and molecular biologists do to study these sorts of questions. Critical for understanding the paper next week. Has anybody heard of position effect variegation before? This is kind of a crazy genetic thing. This bit here is just to demonstrate what position effect variegation is. Sometimes in certain phenotypes, certain traits you can observe, you'll see little differences. Like instead of a red-eyed fruit fly, you'll see sometimes its ommatidia are red, the little segments that make up the eye, and sometimes they're white. It's not all red, it's not all white. It's kind of random looking.
Partial effect.
It's occasional effect, random effect. So what's happening, theoretically, is that there's a gene, well, let's take this cartoon on the left. Let's say the W gene is one that produces white ommatidia. When it's mutant, when it's turned off. So in a normal fruit fly, that W plus gene, wild type W gene, makes red eyes, because most of us know that Drosophila eyes are normally that color. They're not normally white. White's a mutant. But on occasion, what happens to that W gene? If it happens to be on a chromosome where there's been an inversion, that W gene can be moved near to that squiggly line. What's the squiggly line? Heterochromatin, which can inhibit gene expression, right? Heterochromatin, tightly compressed DNA, can turn off gene expression. So in fruit flies, so the top one is the wild type X, as it says there. And this bottom one is the chromosome that produces all of the ommatidia. Some of them are white, some of them are red. What's happening to that W plus gene? It should be making always red, totally red eyes, red ommatidia. It's really close to this region of the chromosome that's heterochromatinized. And so what happens is sometimes, by chance, it's so close that that heterochromatin, the boundary is not as exact as it looks here. The boundary of where heterochromatin stops, and then there's euchromatin genes present. So on occasion, that heterochromatin might spread a little bit farther than it normally does onto the chromosome. So it compresses, it condenses that W gene, turns off gene expression. Then you have white eyes, or white ommatidia. Just because occasionally that gene gets turned off because where it's located in the chromosome. If the gene is near heterochromatin, position effect. The position of the gene on the chromosome dictates whether or not it's occasionally turned off, always turned off, or never turned off by heterochromatinization, the formation of heterochromatin. Okay. What causes this heterochromatin? histone modifications. So that's the connection between SIR2 and position effect. There are proteins that are going to tell histones, you should turn off gene expression. Here's an acetyl group, or here's a methyl group. You should be highly compressed DNA. So it's those little chemical modifications on the histones that cause heterochromatin formation and can cause genes to be turned off or turned on. In yeast, it looks more like it's shown on the right. Normal yeast look like that. Nice, round, white, pearly colony of yeast. That's millions of yeast, like bacteria. Start with one, they divide, they grow, and they make a colony. If you have a colony that's lacking in this particular gene, they don't have that gene, they'll make a red colony. I'll tell you what gene this is in just a second. It's one of the genes you'll read about a lot. But if you move that gene from where it is found there in the top, the euchromatic, if you move that gene closer to the black, the heterochromatin, that position effect happens again. Sometimes it gets turned off, sometimes it doesn't. And this is the type of colony of yeast you see. It's like the red-eyed, white-eyed fly. Sometimes you see red colony, part red, part white. That says that when you see S. cerevisiae that looks like panel C, that means that the gene is near heterochromatin. The position effect variations happening, it must be near the heterochromatin. If it's white, does that mean the gene's on or off? The gene's turned on when it's white, top panel, panel A. And whenever you see it's red, that means that gene has been silenced. It's been turned off. Okay, good. So the equivalent of whatever this black blob there is. Right? There are regions of the yeast that are always heterochromatic. So there are a few loci in yeast that are heterochromatic all the time. You might see these position effects. Those are miting type loci, not surprisingly, because most organisms do this. The centromere is almost always heterochromatic, no matter which species you look at. Telomeres, same thing. Functional DNA, not DNA that contain genes. Telomeres are just there to protect the ends of our chromosomes. They're usually heterochromatic. And also ribosomal DNA loci, which is kind of a strange story. So, in case you've forgotten how yeast mating works, did we talk about shmoo tips at some point in the past? Yeast have two different sexes, right? Unicellular organisms, but they've got two sexes, which is kind of crazy and one of the reasons I love yeast. And the way that works is that there are two loci here on chromosome 3. They're called alpha, or HML alpha, and A. Yeast have two mating types, the A mating type and the alpha mating type. And those two regions of the chromosome are silenced. Those genes never get transcribed from those locations. What happens is that an individual yeast will make a copy, as that arrow indicates. They'll gene convert. Recombination copies one or the other of those, either alpha or A, into the middle of the chromosome, the mating type locus. And that gene then is turned on. So that gene is in euchromatin. And whichever copy of this gene you have, the A type or the alpha type, you make that protein. And that defines what mating type, sex essentially, male or female, A or alpha. So the original loci, HML and HMR, are always in heterochromatin, because it would be really confusing sexually for the yeast if it was expressing both the A and the alpha proteins at the same time. It wouldn't know what sex it was.
What's the point of them having sex?
Because yeast do sex. Did you know that? Yeah, unicellular organisms, but they sexually reproduce. They can sexually reproduce. They do both. Normally, yeast are just dividing in half and making two daughter cells and two daughter cells, but you can actually force yeast to do sexual reproduction. And that's where this comes in. So that's mating. This is just another diagram that says essentially the same thing. Gene conversion, copies one or the other, never both, of those HML and HMR loci. And look what it says up there. SIR gene products silence HML-A and HML-alpha. So it's including SIR2, the protein that we're going to be talking about. These genes do something to the chromatin, to the histones, that keep those regions of the chromosome always turned off. They're always heterochromatin. So that's what the authors are talking about when they talk about the HML and the HMR loci. Telomeres also heterochromatize. Look who's there again. SIR proteins doing their job, presumably doing their job, we'll see, in making the ends of the chromosomes highly compressed, highly compacted. And also in the centromere. Centromeres are highly compacted, heterochromatic. Okay, and ribosomal DNA. Long, complicated story that's not necessary. Okay, last few bits of information. The authors are going to talk about specific genes in yeast that they use to ask whether or not SIR2 is having an effect on gene expression, the absence or presence of SIR2. One of the genes they'll talk about is AD6. It's one of more than 6 genes that are responsible for adenine biosynthesis. What's adenine? How do you know adenine? It's part of the code. It's A in DNA, right? This is really important for yeast to make adenine. Can't replicate DNA without adenine. So there's a crazy thing about yeast that when they're missing one of these genes, they can't make adenine. That also means that one of the precursors to producing adenine accumulates inside the yeast. And that precursor is red. So you can tell when a yeast isn't producing its own adenine because that yeast turns red. So yeast geneticists for decades literally have used this knowledge. Anytime you see a red yeast colony, you know that it's not producing the adenine, the AD6 gene product, the protein.
Is that like a cloning kind of thing, or is it just a natural product? Why they turn red? Is that like...
It can be either a natural mutation or it can be an induced mutation. So researchers can actually explicitly get rid of the adenine 6 gene to make the yeast look red.
I mean, the red effect, is it like...
The red effect is normal. So when you don't have this gene, the yeast normally turn red.
They just turn red?
Yeah, because they can't fully synthesize adenine. And it's one of the compounds that would be turned into adenine, but can't, that is red, that gives it that pigment. Yeah, so it's endogenous. It's something that the yeast would naturally do if they didn't have. If you go out in nature and find an adenine 6 mutant, they'd be red. Yeah, exactly. It's not GFP or anything else. That the only thing that has happened is somehow the adenine 6 gene is either mutated, so it doesn't produce the enzyme, or it's been silenced by heterochromatin. So that's what next week. The other gene that they talk about, or at least use, is URA4, part of the uracil biosynthesis pathway. Right, same deal. It's another nucleotide. But in this case, they can use 2 different tests to ask whether or not the uracil, URA4 gene is present, if it's being actively transcribed and turned into protein or not, if it's been silenced. So there's positive selection. That is, you want to know, do the yeast have the gene? You can grow the yeast on plates that don't have any uracil. So normally, yeast are auxotrophs, most yeast. If they need something, they can take it up from the environment. If they need a biosynthetic molecule, if they need adenine, glycine, amino acids, nucleotides, they can take it up from their environment. So if you have a yeast that doesn't have this gene, URA4, if it's mutant, it can't produce its own uracil, it's fine as long as you put some uracil in its medium that it's growing in. It can take up the uracil and it's fine. So That's one way you can tell whether or not a yeast is turning on this gene. Is it silenced or is it not? If ura4 is silenced, the yeast aren't making this enzyme, can't make their own uracil, can't grow unless you give them uracil. So you compare, here's yeast growing with uracil, here's yeast growing without. They're all going to be fine when you give them uracil. But if you take it away, the ones that aren't making this protein will not grow. So you can that's one test you can use to ask whether or not this gene has been silenced. The other test that they use is that one that says negative. So this positive test means that if you've got the gene, you grow. Negative means if you've got the gene, you die, which is a bizarre thing. You wouldn't normally think that a yeast turning on its normal proteins that are used to synthesize uracil would be lethal to it. But it is lethal when you add this drug, 5-fluoroerotic acid. So if you add that to the yeast's medium, its ura4 gene converts 5-FOA into 5-fluorouracil, which kills the yeast. Poor yeast. So that's a way to tell, that's another way to tell that's totally separate from that positive selection scheme, negative selection. If their ura4 gene is on, they die if you give them 5-FOA. So you'll see examples of both of those in this paper. Growing yeast with or without uracil to ask whether or not the ura4 gene is on. And also growing yeast with and without 5-FOA to ask exactly the same question. It's just the reverse logic. Is ura4 present? You can grow in uracil. You can grow without uracil. If it's present, you die in 5-fluoroerotic acid.